Let's talk of sperm DNA fragmentation. And what does it mean? It refers to a break and a damage in genetic material contained within the sperm head. While a standard semen analysis evaluates sperm count, motility, and morphology, that's a shape, it does not assess the integrity of the DNA fragmentation index, is a specialized test that quantifies the percentage of sperm with damaged DNA, offering a deeper insight into male fertility potential. Now, the DNA inside a mature sperm is tightly compacted, and the cell has minimum cytoplasm and has limited capacity to repair any DNA defects. This makes it highly vulnerable to damage, particularly from oxidative stress caused by imbalance of reactive oxygen species or free radicals. What are the clinical implications of high DNA fragmentation? Now, the integrity of the sperm DNA is crucial for successful conception and healthy offspring. And while when the level of DNA damage is high, it can overwhelm the egg ability to repair the defects after fertilization. What we do know is that elevated levels of sperm DNA fragmentation is associated with several negative reproductive outcomes. Unexplained infertility, where sperm count is fine, but the DNA fragmentation may be higher and damage. Failed assisted conception, where high sperm DNA fragmentation is linked to lower success rates with treatment like intrauterine insemination and in vitro fertilization. Now, you can also see poor embryo development, which can lead to poor fertilization, leads to poor growth of embryos from day 3, and failure to develop blastocyst stage. And finally, there is a link to recurrent pregnancy loss, where there seems to be a high association between high sperm DNA fragmentation and the increased risk of miscarriages. Therefore, it's worth considering a sperm DNA fragmentation test in individuals or couples experiencing these issues, or those with risk factors such as varicocele. Now, why does the sperm DNA get damaged? And it can arise from factors from inside as well as from outside the testis. There are problems during sperm production. There are errors during process of sperm maturation, including defective chromatin packing, can lead to production of sperm with damaged DNA. Post-testicular damage. When sperm leaves the testis, it gets stored in the epididymis before ejaculation. The longer they remain there, the more susceptible they are for damage from oxidative stress. And finally, life and environmental factors. Choices. These environmental factors are major contributors. This can include obesity, smoking, excessive alcohol use, poor diet, exposure to environmental toxins and pollutants, and certain medical conditions such as health issues, varicocele infections, chronic inflammation, poorly controlled diabetes, and high fever. So this is something which is often forgotten. Finally, we come to advanced paternal age, and the risk of sperm DNA damage naturally increases as men get older. The management and treatment of high DNA fragmentation is aimed towards mitigating the underlying cause. One, lifestyle changes and antioxidant supplementation. For men whose high DNA fragmentation is linked to lifestyle factors or treatable conditions like infection or varicocele, the first line involves treating those. Cut down smoking, reduce alcohol, adopt a healthier diet, exercise, resistance training, mainly to increase the testosterone levels, oral antioxidant therapy for at least 3 to 4 months can be effective. Supplements such as containing vitamin C, E, coenzyme, and selenium may help to neutralize free radicals and lower sperm DNA fragmentation. Optimize ejaculation frequency. Shorter periods of sexual abstinence can reduce sperm DNA fragmentation. Frequent ejaculation ensures that the sperm spend less time in the epididymis, minimizing the exposure to damaging oxidative stress. For couples undergoing fertility treatment, a shorter abstinence period for around 24 hours before producing sperm sample may be recommended. Testicular sperm retrieval. In some cases where DNA damage occurs predominantly after the sperm leaves the testis, if persistently high DNA fragmentation is found despite other interventions, it may indicate a post-testicular problem, such as then removing the sperm from the testis by a surgical procedure may improve the results and allow us to perform ICSI. Now, techniques such as using zymote preparation may also help in choosing the sperm with the least DNA fragmentation and help in improving the success rates of IVF. Again, there is research saying that it is time which we start looking at men more closely in terms of IVF treatments, and probably that is true.